Avila, and I will be presenting the work titled A Lasso QSAR Model for Predicting Caco2 Permeability in Medicinal Plants from the Biodiversity of Peru. The intestinal absorption can be described in vitro using a Caco2 model, where the value obtained is the apparent permeability, which describes the transfer of a substance through a cell barrier. This model is useful because with this value we can determine the oral bioavailability, the human intestinal absorption, and this is related to the efficiency of the drug. Considering this, our goal in this work was to develop a QSAR model that links the structure of known chemical compounds with their known apparent permeability value, also known as log-PAP, using three methods: multiple linear regression, partial least squares, and least absolute shrinkage selection operator, also known as Lasso. Once we had this model, we used it to predict the apparent permeability value of compounds found in Peruvian biodiversity. We began by obtaining the structural information of 1,817 chemical compounds with known apparent permeability values. We calculated the molecular descriptors of these chemical compounds, where we obtained 3989 molecular descriptors. Then we divided the dataset into fractions. 80% of this dataset was used to develop the models. This information went through preprocessing for feature selection, reducing the molecular descriptors to 188, with which we developed three models: MLR, PLS, and Lasso. Once generating the models, we evaluated them with the remaining 20% of the dataset. We evaluated the effectiveness of the model, obtaining parameters as RMSE and R-squared. As a result, we obtained three models with similar values of these parameters, and the one with the best results was the Lasso model with a value of RMSE of 0.45 and R-squared of 0.65. Also, using the Lasso model, we found these 5 molecular descriptors to be the most important. We applied the Lasso model to obtain the apparent permeability value of 1,041 compounds of Peruvian biodiversity. In this figure, we can see the distribution of this predicted value, and we found that 59% of them showed an apparent permeability value greater than -5. So these compounds could have a high intestinal absorption. Between these compounds, we found 9 flavonoid compounds related to anti-malaria, antimicrobacterial, antifungal, and aldose reductase inhibitory activity. In conclusion, the present study employed 1817 molecules to build the models, taking into account the RMSE and R-squared values, and it was determined that the Lasso model showed the best performance. It was determined that the log-PAP values are influenced mainly by these 5 molecular descriptors, and the QSAR log-PAP model determined that 59.16% of compounds from the natural product database have a log-PAP value greater than -5, therefore they could have a high intestinal absorption and a potential high drug bioavailability. Finally, I thank the Vicerrectorado de Investigación y Posgrado for the financing of this work, the Instituto de Investigaciones de la Amazonía Peruana for lending us the supercomputador Melanie to perform the calculations, and you for the giving opportunity to present this work. Thank you so much.